So, very good morning to everyone. It's my immense pleasure to welcome you all to the Altum platform for an sixth session of an inspiring woman in scientific academia. Uh, Speaking about a women, encouraging a women scientist is a crucial for fostering a diversity, innovation, and progress in the scientific community. By providing the support, resources, and opportunities, we can empower women to pursue their passion and contribute their unique perspectives to achieve their full potential in the scientific field. Today, I welcome one of the esteemed women scientists, Dr. P.L. Sujatha ma'am, to the Altum platform. And we extended an heartfelt invitation to you to join us and share your groundbreaking research, innovative ideas, and inspiring journey in the world of science. To speak about our today's speaker, ma'am had 23 years experience in working with an Tamil Nadu Veterinary and Animal Science University. Ma'am had played a pivotal role in guiding the research scholars, serving as a chairman or a member of an advisory committees of various levels from a level of PhD to undergraduate, had teaching expertise spans from a undergraduate, postgraduate, including a BTech in food technology, MTech in food technology, PG diploma in bioinformatics, and PhD in intellectual property management. And she has developed the practical and instructional manuals and introducing innovative methods as a computer assisted instructions and computer based trainings. And to accolade to her achievement, she had a lots of national and international publications. And it's my immense pleasure to invite Dr. P.L. Sujatha ma'am to the Altum platform to represent today's sixth session of an inspiring women in academia. And I wholeheartedly welcome you to your platform, ma'am.
Yeah. Thank you, Engineer Divya, for your brief introduction. And yeah, very good morning to one and all who have joined in this virtual meet. Let me take this opportunity to thank Altum Technologies and especially to Dr. Sakshi, Engineer Divya, for giving me a chance to meet all of you and to share our knowledge in designing a database on bioinformatics. So our research work was on designing and development of bioinformatics database on gastrointestinal disorder of bovines. So the word bovine comes from the Latin word for cow, though the biological family called the Bovidae, which actually includes not only cows but also oxen, goat, sheep, bison, and buffalo. So, as you all know, the GI tract of bovine, it starts from mouth and then runs to esophagus, and then it goes to stomach, which consists of four parts such as the rumen, the reticulum, the omasum, and the last one is the true stomach or the abomasum, and it goes up to small intestine, large intestine, and ends with the anus. So the GI disorders of bovines are of two types. One is infectious, which is caused by the microorganism like virus, bacteria, fungi, and parasitic organism, and non-infectious such as acidosis, bloat, etc., or overeating of carbohydrates, etc. And the currently we treat the GI disorder of bovines with the help of antibiotics, electrolyte therapy, supportive treatment, and fluids. And we have vaccines also for some of the GI infections. And as a result of continuous usage of antibiotics, the animal will develop resistance against this antibiotics. Apart from the antibiotic resistance, the antibiotic usage might result in severe side effects. And as you all know, people are now concentrating on the usage of herbal medicine as an alternative to the allopathy medicine. So, similar to the human herbal medicine, the veterinary herbal medicine involve the plant-based medication in animal health care under therapeutic, prophylactic, or diagnostic application. In human health care or animal health care, the implementation of herbal medicine has a long history, which you can trace back over millennia. The prevalence of infectious disease in bovines has a significant impact on the development of cattle industry and public health security. So the identification and analysis of the pathogens are very important for the infectious disease research. And however, the existing databases pay very little attention to the bovine diseases, and the data is almost scattered in different databases that is available in the databases. So there is a need for the effective integration, analysis, and visualization of this type of data, along with a sort of bioinformatics analysis, which might help the scientists to develop a novel lead compound for the treatment of the diseases. And apart from this, the increased costs associated with the vaccination and disease treatment can also affect bovine breeding. So we are living in the world of big data. Open information and resource sharing or trending in the era of big data. The integration of these different types of pathogen along with its treatment can enhance the data reusability and accessibility. And however, there are databases which are scattered in the internet on the types of bovine breeds or the type of infectious diseases, but and the type of treatment, but there is no single database in the internet which talks about the treatment using plant principles. And recently, a database on sheep and goat pathogen database was developed by the School of Tropical Agriculture and Forestry in China. So this database holds information about the pathogens which affect the sheep and goat species. So we decided to create a database on the bovines, which can hold data about the GI disorders along with the plant principles, which might against this GI infections. So these are the objectives of our research work to collect and listing out of GI disorders of bovines, to identify the active medicinal herbs used for the GI disorders of bovines, and to perform the molecular interaction between the drug targets identified, and to carry out the simulation of the best doctor complex. And the final one is to design and develop the bioinformatics database on gastrointestinal disorder of bovines and host it in the website. So this database contains bioinformatics data for infectious disease of the GI tract. In addition, it holds information about the Indian medicinal plants which might act against the GI disorders, information about the bovines, and information about the gastrointestinal disorder. And this BIDGIB, that is Bioinformatics Database of Gastrointestinal Disorder of Bovines, it gives an in silico approach in drug discovery for GI diseases. And this database could be useful for the researchers and scientists to find novel drugs to combat GI infection in bovines. So this is the materials and methods we followed for this research. We first reviewed and collected data from the online sources containing infectious diseases and pathogen data, and identified some specific databases such as NCBI, DDBJ, UniProt, and EMBL as candidate sources. We also conducted interviews with the scientists and veterinary practitioners who work on this bovine pathogens, and identified some high-level findings such as the purpose of using the data, the type of information that should be available in the database, and how users can obtain pathogen-related information, etc. And this insights were used to generate what you call the user-centric data set of bovine pathogen and disease information. The GI disorders of bovines are collected from the mainly from the literature and databases and interview with the scientists. So the causes of disease, infection, clinical signs, diagnosis, and the current treatment, everything was studied. And the next step is the Indian medicinal herbs, which might act against this GI disorder, was collected from the literary sources. Ayurveda and Siddha knowledge, and also with the help of veterinary practitioners, we collected the information. And then the protein targets, which are responsible for the pathogenesis of each GI disorder, are identified with the help of databases like Uniprot, NCBI, and literary sources. And then the next step, the structure of this target protein was retrieved from the PDB. The structures which are already available in the PDB was retrieved, and the structure where they remain, the structure which is not available was modeled using ITAS, that is iterative threading assembly, and also Discovery Studio. And the structure was validated with the help of the What If server and Discovery Studio. And ligands, we retrieve the ligands from the impact, that is Indian medicinal plants, phytochemistry and therapeutics, and PubChem and Dr. Duke's phytochemical and ethnobotanical databases. In some cases, we are not able to get the STF format of the ligands. In those cases, we just used ChemSpider to draw the STF format with the help of the chemical formula that is available in the documentary sources. And the molecular interaction was performed using Discovery Studio. And then we carried out the pharmacokinetic analysis and pharmacodynamic analysis for the best dot complex. And finally, the database was developed using MySQL as the backend, and .php and .NET as the scripting language, and hosted in the Apache web server. And this slide is just to show the IT components used for the creation of the web database. And we preferred using MySQL as the backend because of its data security, data integrity, and query flexibility. And we used PHP and .NET because, as you all know, these two are the most widely used scripting language in web application development. And PHP and JavaScript is compatible with MySQL. This is just the script we use for the creation of the database. And then we are moving into the results. So we just identified the infectious and non-infectious disorders in the bovines. For the infectious, we identified four types of infectious diseases. For example, viral infection. The best example for the viral infection in bovine is infection caused by rotavirus, and bacterium infection caused by cryptosporidium or salmonella, and fungal infection caused by aspergillus fumicates. And the last is most neglected area in GI disorder of bovine is parasitic infection. So this GI tract of bovine is infected by variety of parasites, particularly helminthes, which causes clinical and subclinical parasitism. Clinical means the animal is infected and the symptoms are clearly visible. And subclinical is the animal is infected but the symptoms are not visible, and the infection could be identified only with the help of laboratory techniques. So these parasites adversely affect the health status of animals and cause enormous economic losses to the livestock industry. And the GI parasites not only affect the health but also affect the productive and reproductive performance of the cattle and buffalo. And the parasitic infection results in the considerable losses in milk production of cattle and buffalo. And the non-infection, as I said, we just try to study the non-infectious infections such as bloat, fatty liver, indigestion, abdominal distension, cirrhosis, etc. For the infectious, we identified 40 targets from the different sources. out of which 19 structures we downloaded from BDB and 21 we just model using the ITSR and Discovery Studio. So we concentrated more on the infectious rather than non-infectious because non-infectious, after some point of time, the animal restored back its original condition, except very few conditions like fatty liver. So we downloaded some minimal targets. The targets were not available. In fact, we downloaded minimal targets for fatty liver. So this is just the table which shows the condition, pathogen, and the type of a disease. So in the ligands, we just try to scan through the literature and different. We discussed with different farmers who own the bovines to check the type of herb or the herb they use for the treatment of different disorders. And we downloaded the STF format from different databases, and we identified around 3,769 ligands which might act against the GI infection of bovines, and we filtered from 3769 to 2779 based on the Lipinski rule of 5. So these are the different types of bioinformatics data that is available in the database. The target data, we have the name of the target, the number of active sites in the target. If the structure is available in the PDB, then the PDB ID will be there. Or if it's a modeled structure, UniProt ID will be there. And if it's a modeled structure, the validation data will also be there in the database. And the secondary structure of protein is available. In the case of ligand data, we have a plant name, the ligand name, the PubChem ID, or a PubChem ID or IMPACT ID, molecular formula, the SMILES format, and the structure of the ligand. And the docking table, we tried the docking with the libdog. So the libdog score will be the hydrogen bonds, RMST value, and the energy. So we have some images like the 3D interaction of the receptor and the ligand, and 2D interaction of receptor and the ligand, and PK analysis result data, and PD analysis result data in the database. So this is just an example to show what type of data will be there in the database. This is for the bacterial infection in the bovines. So these are the different bacterial diseases, and there's a plant's inhibitor. So this is the Lipdog score and the phytochemical and the name of the Andrographis paniculata. And this is for the viral infection, and we identified different viral infection in bovines and the plants. And this is the just the type of this is for the fungal infection. Interestingly, the Andrographis paniculata has an activity in all the bacterial infection or viral infection or fungus. So it's a well-known fact that Andrographis paniculata is an immune booster, which boosts your immune activity. So we also have the um, pharmacokinetic result in the database. So far we have uploaded data that is obtained through SwissArmy and TalkStree. We are currently working on a Discovery Studio, and we'll upload it in um, future. And we also carried out the pharmacodynamic analysis of the ligands with the help of the PASS online server that is available in the Way to Day Drug website. So we identified whether the compound has antiviral property or the antifungal property. So this is actually the website of the homepage of the database, bioinformatics database on gastrointestinal disorder of bovines. Let me just show a short demo of the website we have created. Um, can you able to see the website, Divya? Yeah, thank you. Yeah, this is the website or the database we have created to hold the data on the gastrointestinal disorders of bovines, along with some bioinformatics analysis. So this we can do a general search here or with the menu search. Let me just show you the general search. You can search using a bovine keyword or the plants which might act against the GI disorder or the type of infections. So I'm searching through a breed, and Burgor is actually a breed. So you can see the information about the animal picture, its characteristics, its distribution, and most importantly, the references. That is where we have collected this information. And the plants, the Osimum bacillium, the plants which might act against the GI disorder. So this database holds the plants which might act against the GI disorders and not all the plants. So if you click on the plant's name, you'll get the what's the vernacular name with the taxonomic classification, the description and where it is distributed, what is the usage, and finally the references. This is the general option, or you can click in the menu wise option. You can select the first one is bovine breed. I am just clicking on the Gir. Gir is a breed native of Gujarat. It's the high milk yielding breed in India. So it's the name of the breed, its characteristics, its distribution, and what are the other names and references. The breed data mostly we selected from the ICR websites, because ICR has a standard database which holds data on the animal genetic resources, and we collected data mostly from the ICR website on the Indian bovine breeds. And this is about the GI disorders. So once you click on the GI disorder, it will ask for the, I mean, what type of GI disorder you are interested in, whether it's you're working on the infectious or non-infectious. Let me search infectious. Infectious, again, you'll get one more query box. What type of infectious diseases you're looking for, whether you're working on the bacterial disease or fungal disease or viral disease or parasitic or protozoal. Let me search bacterial disease. So again, what type of disease? Let me search for salmonellosis. It's caused by salmonella. So the picture of the infected animal will be there. What is the name of the disease? What are the characteristics? And what is the cause for this particular disease? What is the treatment currently followed in the country? And the references. Or I'm interested in the viral. So let me check rotavirus. This is for the virus, name of the disease, and the characteristics. And similarly, as I said, causes, this is the picture of the infected animals. Rotavirus causes severe diarrhea, and treatment and the references. And similarly, I'm working on the non-infectious. Non-infectious, let me search for bloat. Yeah. So it's just a gas accumulation of gas. Bloat is accumulation of gas. So name of the disease, what are the characteristics, what is the treatment. So this is the picture of the infected animal. And next, the plants, what type of plant which can act against this GI disorders. So let me check for Cissus quadrangularis. So this Cissus quadrangularis is used for GI disorders. So what is the botanical name? What is the common name? This is a picture of Cissus. What is the taxonomical classification and its distribution? And what is the usage? And finally, the references where we have collected the data. And this is the most unique feature of this database. So this three types of data, you can get it in any database that is available in website. I mean, Indian bovines, there is a standard database on Indian bovines, and GI disorders, there is a database. And Indian medicinal plants, of course, we have Impact or Dr. Duke's ethnobotanical database, and so on. And this is the unique feature, that is bioinformatics data. So, infectious diseases. So, for which disease you are searching for a lead molecule to find a drug against? So, for example, I'm working on viral diseases. So viral diseases, again, rotavirus. I am planning to identify a new drug molecule against rotaviruses. So once you select this option, the database will show you what is the target, how many active sites are there in the target, and this is the modeled structure. So Uniprot IT is there. So this was, developed using the IT, sir. So compound this compound from the Tinospora cardifolia has a very good interaction with this capsid protein. So the score was 112 and hydrogen bond is 6, and this is the binding energy. This is just an 2D and 3D interaction images. And you'll have the pharmacodynamic result. See this compound has antiviral activity. Antiviral 85 percentage. This compound has antiviral activity. So you can very well judge this compound has antiviral activity, and it can fight against the infection caused by the rotavirus. And you have Swiss Army result and toxicity profiling. So this a sort of prediction. The scientists can use this compound to identify a new drug molecule against this rotavirus infection. Similarly, if you are working on the bacterial diseases, say enterotoxemia, this Andrographis paniculata. So this plant, and particularly this compound, dihydroandrographylline, has a very good interaction. See, the score is 142. It has a very good interaction with this toxin, epsilon toxin. So, epsilon toxin is the target which causes the toxicity in the animals. So, which you can use this compound to identify a new drug molecule, and then you can see the image, and you can see the what do you call the pharmacodynamic activity, and at the end you'll see the toxicity profiling also. So this is the general menu search, or else you can use you can search any term using this is similar to your the search box which you find here. Both this or the have the same option. So we are still working on this database. We are just updating it. We are carrying out the molecular simulation work to identify the flexibility of the molecule. Sorry. farmers and the veterinary practitioners. So, what is the uses and uniqueness of this database? So, you can find database on the bovine diseases or the medicinal plants databases and the bovine breeds databases are available. When you compare those databases with this BIDGIB, it's a unique one among those databases. It contains information not only about Indian medicinal plants, Indian bovines, and GI disorders of bovines. It also contains the bioinformatics data of infectious disease, along with pharmacokinetic and pharmacodynamic properties of identified lead molecule. And you can, I mean, the researchers can use this data available in the bioinformatics database of GI disorder of bovines to find a novel drug candidate. And also it could be useful for students to fetch information about especially for the veterinary practitioners or students who are in the field of veterinary and animal sciences can find information about the different bovine breeds that is available in India, and also the medicinal plants and gastrointestinal disorders. So we work independently in bioinformatics to identify drug molecule using the plant principles for different diseases or disorders. And what we generally do, we just try to submit our project or thesis report, or in some cases we used to publish. So what I request is, in addition to the publication of your research work using plant principles, you can also think of creating a database where you can upload your data and people working anywhere in the globe can use your data and create a new drug molecule. So you can also avoid the duplication of duplication of the research. If the plant principles has been studied in some other part of the globe for the same disease, you can think of some other plant principles which might work better than what is already reported in the database. The future plan and what we are planning for the simulation and molecular dynamic study to prove its prove its efficacy. And we are also planning to do the in vivo and in vitro studies. It will take time, but because the database hosting is a continuous work, it is not one-time work. We need to update it, and we need to find more molecules, more targets to help the practicing veterinarians. And this publication, there is a publication already there in the net on the current bioinformatics on the identification of novel plant-based drug candidate against Jones disease. We just published article on only one particular disease, that is paratuberculosis or Jones disease, in the current bioinformatics. So I conclude with my presentation. As I said, this database will give you an in silico approach in drug discovery, and one can use the data that is available in this database to identify new drug molecules to drug molecules to combat gastrointestinal disorders in bovines. And as I said, I request those who work in the field of plant principles to create a database, and you can also, I mean, share your data with scientists working in other part of the globe. And thank you for the opportunity given by the Altum Technologies. Thank you, Dr. Sakshi and Engineer Divya and our dedicated team for giving me an opportunity to share our research work in this virtual platform. Yes.
Yeah, ma'am. I think it was an inspiring presentation. I think it's an another domain of veterinary research, but people who are
Thank you.
looking for how you can integrate animal informatics, especially you can say veterinary informatics. It's one of the more growing domain where people have to understand only doing the veterinary research or not going to help them or give the hand along with the in silico. Definitely they can see the accelerating research with a shorter span of time, and they can have a fruitful results, I could say that.
Yes.
Thank you, ma'am. I think there are few questions are there for you. I think they feel so excited to answer, take your
Yeah.
questions. And there is a question. You can you able to see that, ma'am?
Yeah. Is the database publicly accessible?
Access.
Yeah, at present it's no. So we are planning to host it in the Tanua's website.
Hmm.
It might take, say, around 3 to 6 months. We need to get official permission from our university authorities. So you can check Tanua's website after 3 or 4 months. It will be publicly accessible.
Oh, okay. Thank you, ma'am. I think once it is publicly accessible, that everyone across the no, I won't
Yeah.
don't say across the country, across the world, people can understand the importance of the database because you showed the integration that how bioinformatics of an, a plant, in I could say the plant and animal and how the disease are integrated together, where people can understand more about the plant and their byproducts, how to treat for various bovine disorders. And going
Yeah.
on, I think there is one more question you can see, ma'am.
Yeah, the second question was on how users can benefit by utilizing this database. So, as I said, you don't need to do the molecular interaction or simulation work. You can directly take this compound to the next level of drug discovery. You can directly take this to in vivo or in vitro studies. So, this database will help to reduce your time frame in identifying a drug molecule for GI infection.
Oh, that would be great, ma'am. I think that is really required. It seems without
Yeah,
we haven't anything in our hand, it makes more easier for the
yeah,
people
that's
to
right.
go
That's
on.
right.
I think so. That's right. And there is one more
Yeah.
question, ma'am. You can see there.
How unknown targets or model. Yeah, as I said, and previously we used the portal iTaser because that time we don't have the license for Discovery Studio for the modular protocol. So recently, say 1 year back, we got the modular protocol license from the BioVia Discovery Studio. Nowadays, we are using modular protocol of Discovery Studio.
I think that would be great. I think knowing the unknown targets modeling is tough. I think that can be done by using Discovery Studio very well. And there is one more question, ma'am. You can see that.
Yeah, how bioinformatics will benefit in veterinary research. You cannot separate veterinary from human. Whatever benefits we get from the bioinformatics, the same benefit the animals also can get from the bioinformatics. You can identify new vaccination, new epitopes. You can identify it with the help of bioinformatics tools, and you can identify new drug molecules. The same way, animals also can be benefited. You cannot separate human from animals.
That's great, ma'am. That's great. Thank you. I think there is one more question.
Yeah. How templates are validated during modeling because modeling of animal targets or tasks. Yeah, I explained it during my presentation. The templates are validated using the modeler website. We had an issue when we tried modeling using iTaser. So nowadays we are not facing this issue because of BioVia.
Yeah. Yeah, I think template validation is very important before you start
Yeah,
doing
that's
modeling. Yeah, I think a Discovery Studio will pay a better way and best
yeah,
way
it's
for
a
an
better tool
people.
than your iTaser or modeler.
Yeah, that's true. Yeah, yeah. I think, yeah. Another question.
Yeah, we tried with individual herbs. We are not tried for the synergistic activity of the herb. We tried for the individual herbs. Maybe it's this is an we can try this also. Synergistic activity also we can try. Thank you, Ms. Giti Krishna Sahu. Thanks for this idea.
Yeah, good. Thank you. I think there are interesting questions. People want to know how the polyherbal formulations are very important for treating the treatments of bovines and so on. And thank you for asking that question.
Yeah, if any
You have
queries, it can be addressed to my email. I'm ready to answer the queries.
certainly, yeah. Any other question? I think yeah, it's. I think there is always applause because this presentation, I think, very unique, and people will understand. That's what you told. There is no differentiation for an animals, or bioinformatics can be applicable at any domain wherever we want to integrate it. And I think it's very informative sessions. And thank you for answering. It's our pleasure and amazing sessions. Yeah, ma'am, you can see over there. And
Thank you. Thank
one second.
you for
One
giving me an
second.
opportunity. Thank you.
Posted. 1 second. Ma'am.
Yeah.
So to add a value to your presentation, and Altam was really very inspiring to give an e-memento to you along with the presentation. I think 1 second. I think they are posting it. Yeah. So thank you once again, ma'am. We'll come and reach you out with this one. And then further, before concluding, I would like to take at the end, we are come to the conclusion. I extend a warm, I think, thankful to all the women scientists who has been joining this Altum platform as we are celebrating women's achievements in sciences. And today's presenter, Dr. P.L. Sujatha, ma'am, your expertise, contributions, and your voices for an bioinformatics with veterinary are invaluable, and then promoting diversity and excellence in the innovations in STEM fields. And let's together inspire the next generation scientists and drive the positive change in the field and empower and uplift, and we showcase the remarkable contributions to the women's science. And thank you so much, ma'am, and thank you for taking our invitation and joining with us. And it's a very immense pleasure to have you in the platform in the Alchem, where we are celebrating a women's scientist achievements in the inspiring scientific community. And further to all the tuners, and stay together and let's pay a brighter and more inclusive future. And welcome to the Altum platform once again. And stay tuned for we have two more sessions are left out and catch you up. And once again, I thank PL Sujatha ma'am for having
Thank you.
joining with us in your busy schedule. Thank you, ma'am. Thank you, everyone. Stay tuned and we catch you up soon. Thank you all.
Thank you. Thank you.